So in this video we mainly discuss about the HLA, MHC, major histocompatibility complex. This all topics comes under the serology and immunology, that is BVMLT-201. First of all, we see what is the HLA. Actually, this is a human leukocyte antigen, and this HLA is present in this chromosome number 6. What? This is a chromosome number 6. Already you know that human having a 23 pair of chromosome, right? So in each human having 23 pair of chromosome, and this 23 pair of chromosome number 6 having this HLA region. What? Chromosome number 6 having this HLA region. See, this is the chromosome number 6, and this chromosome number 6 having this HLA region, and this HLA region having the class 1st MSC, class 2nd MSC, and class 3rd MSC, right? So we discuss in this video mainly this HLA and this class 1st MSC, class 2nd MSC, and class 3rd MSC, and how this MSC will be work against the antigen, and how they are help in recognition of self or non-self. Okay. So now we start. So first of all, introduction. What is the HLA? Actually, HLA is the major histocompatibility complex. What? It is the major histocompatibility complex. A human leukocyte. It is the human leukocyte antigen, and it is important part of immune system, which controlled by gene located on the chromosome number 6. Already we discussed that it is present on the chromosome number 6. And it mainly help in the recognition of self or non-self, right? It encodes cell surface molecules specialized to present antigen peptides to the T cell receptor on T cell, an important part of the immune system, and is controlled by the gene located on the chromosome number 6. It encodes cell surface. What? It encodes cell surface molecule. What? It encodes cell surface molecule that is specialized to present antigenic peptide. What? They is specialized to present antigenic peptides to the T cell receptor, on T cell, an important part of the immune system, and is controlled by gene that is located on the chromosome number 6. And it encodes cell surface molecules specialized to present antigenic peptides to the T cell receptor on T cell. This MHC recognized by the T cell receptor, not B cell. Remember this. There are two types of cells, T cells and B cell, right? This MHC recognized or encode cell surface molecule that is specialized to present antigenic peptide to the T cell receptor, So the scientists who discovered first this rejecting transplants or histocompatibility are the George Snell. George Snell in 1903 or 1996 discovered the first component, the MSC, through their role in rejecting transplants in mice, and created the word histocompatibility. What? They create the word histocompatibility because when they do the experiment in which some other organ or cell when placed in mice instead of then their body reject that organ, means they reject that transplantation. If mice having heart or lungs and they transplanted by same organ by the other mice, then the mice in which this transplantation will be takes place, this body of that mice will be rejected. Then George Snell said that created the word that is histocompatibility. that histocompatibility means there is something in the mice or in the body which not accept same organ, which not accept the same organ from same organism, right? So in every organism there is a histocompatibility which when they detect this is not our self or this is not their organ or their cells, then they reject. And that what is created the word that is histocompatibility. Around a decade later, Jin's 1916 to the 2009 uncovered the first compatibility antigen in humans. Experiment by Baruch in the 1970 provided the first indication that immune reactions and control by the MHC. There is the first indication which of the immune reaction that is controlled by the MHC gene. What? That is controlled by the MHC gene. That is immune response gene. Right? And this MHC gene are located on the chromosome number 6. See, this is the normal cartoon picture in which there is MHC. We discussed about the indicating the MHC or T cells and intact antigen, right? So T cell do not recognize intact antigen. Simply T cell not detect the antigen. They takes help from the MHC, right? They takes help from the MHC. See here, this is the whole chicken. This whole chicken is the intact antigen. So T cell do not recognize intact antigen. Interact only with antigen fragments, that is peptide. Peptides are only means this T cells not interact with antigen, means this T cell recognize this antigen via a peptide, only a single chain of peptide, right? So peptides are only recognized when they are associated with self MHC. When they are associated with self MHC molecule that is presented by in this vector, right? Peptides are only recognized by the self MHC molecule. So there are some terminology in this HLA. There are some terminology in HLA. When we discuss HLA, we have to remember this all terminology like histocompatible. Histocompatible means transplanted tissue is successfully accepted as self. Histocompatibility antigen rejection. Histocompatible means accepted, successfully accepted, right? It's okay for our body. Histocompatibility antigen means there is rejection of foreign tissue is the result of an immune response to cell surface molecules. Histocompatibility complex, a region of multiple loci that play major roles in determining whether transplanted tissue is with histocompatibility or in histocompatibility, right? Histocompatibility complex. What is histocompatibility complex? A region of multiple loci. What? It is the region of multiple loci that play major role in determining whether transplanted tissue is with histocompatibility or in histocompatibility. Terminology 2. There are the major histocompatibility. There are two types of histocompatibility: major histocompatibility and minor histocompatibility complex. So in major histocompatibility, what happened? They represented by capital M, right? In minor histocompatibility, they represented by small m. Okay. So in MHC, in major histocompatibility complex, the organ that are transplanted from other organ means transplanted by other individual that are rapidly rejected. Means if they are transplanted, like if my lungs were not working proper, and that lungs were transplanted by another individual, then my body rapidly rapidly reject that lungs. That is known as major histocompatibility complex. So during transplantation, MS, take the MS, doctor consider this MSC, right? In minor histocompatibility complex, slowly they reject the graft, not rapid, right? So HLA, next is HLA, human leukocyte antigen, that is MHC antigen in human, right? MHC antigen in humans. H2 MHC antigen in mice. H2 is represent the MHC antigen in mice. See this. This is the chromosome number 6, right? This chromosome number 6 contains the region of HLA, that is human leukocyte antigen, and this HLA region contains this all three types of MSC, that is MSC first, MSC second, and MSC third, right? So this HLA complex spans 3.5 million base pair. What? 3.5 million base pair on the short arm of chromosome number 6. Short arm of chromosome number 6. HLA complex is in human chromosome 6. This is the alpha beta chain in MSC region, right? This is the MSC 1, that is BCEAGF. This is MSC third, that is C4BFC2. MSC second, DP, DN, DM, LMP, TAP, DU, DQ, and DR. These are all are the sequence. These are the gene structure of human MSC. What? These are the gene structure of human MSC. How the human MSC are look. So first, classical versus non-classical. In classical MSC, present peptide directly to T cells. What's happening in classical? The peptide directly interact with T cells. Suppose this is the T cell and this is the antigen, and this is the antigen and this is the peptide. This peptide directly interact with this T cells. So this is the classical, and this is highly polymorphic in nature. In non-classical MSC, not directly bind to peptide, and this is not highly polymorphic. So classical HLA. In classical HLA, class first, HLA A, B and C. In class first, HLA A, B, C encode the heavy chain of HLA class first molecule. This represent the A, B, C represent the alpha chain, that is the heavy chain of the class first molecule. And this is the class second molecule, that is HLA DP, DQ and DR. DP, this is the DP, DQ and DR. Each has a and B region. See, all having the alpha and beta region, right? So both having the loci of A and B, and this encode the alpha chain and beta chain. What? This encode alpha chain and beta chain of HLA class 2 molecule respectively. Clear? Class 3, various secreted protein with immune function in inflammation, that is TNF, C2, C4, complement factor C2, complement factor C4. So the main function of class 3 is in the secretion of the protein which help in inflammation, and other non-classical gene and molecule, that is HLA, D and CD1. This is the structure of MSC molecule domain. See here. This is the membrane distal domain. This is the membrane proximal domain, transmembrane segment and cytoplasmic tail. So this is alpha 1, alpha 2, alpha 3, right? In class first, there is no beta 1, right? Only beta 2 microglobulin are present. So here alpha 1, alpha 2, alpha 3, and this between the alpha 1 and alpha 2, this is the peptide binding cleft. And in this peptide binding cleft, antigen comes and bind with this peptide binding cleft, and the processing of the antigenic peptide will be takes place, right? So alpha 1 and alpha 2 are the region where peptide will come and bind, right? And this is the class 2 molecule which contain a reason alpha 1 and two types of chain are present in this. That is alpha chain and beta chain, right? And this alpha chain having alpha 1, alpha 2 and beta 1 and beta 2, right? Beta 1 and beta 2. So what happened in this? This is the cytoplasmic tail which inverts or which cross this transmembrane segment. So in this class first molecule, only alpha chain will be cross the transmembrane molecule, whereas in class 2 both alpha and beta chain are present, and these both cross this transmembrane segment. So class first and class second molecule both are membrane bound glycoprotein. MHC are membrane bound glycoprotein, right? Both are membrane bound glycoprotein containing external domain, a transmembrane segment and a cytoplasmic tail. Similar in three-dimensional structure, both having a peptide binding cleft, but class first having alpha 1 and alpha 2 peptide binding cleft, whereas second having the beta 1 and beta 2, right? And two immunoglobulin like domain, membrane proximal domain are present. MSC class first molecule. Alpha chain plus beta 2 microglobulin. What? Two types of chain are present, that is alpha chain and beta 2 microglobulin. Alpha chain are polymorphic and they cross the transmembrane. What? They cross the transmembrane, right? Polymorphic means they present in different form, right? So this alpha chain are present in different form like alpha 1, alpha 2, alpha 3. So this alpha chain are in polymorphic in nature. So alpha 1 and alpha 2 domain form peptide binding cleft, alpha 1 and alpha 2. See, alpha 1 and alpha 2 having a peptide binding cleft, and alpha 3 interact with CD8 on the T cell. This is the T cells, right? So alpha 3 interact with CD8 on the T cells. Beta chain, no polymorphism because only beta 2 are present, right? Only one form of beta chain are present, that is beta 2. So there is no polymorphism in beta chain. So no polymorphism. One folded domain stabilized class first molecule gene on chromosome 15. What? Stabilized class first molecule on the chromosome number 15. Expressed on almost all nucleated cells. Remember this. MHC class first molecule are present in all nucleated cell. Presentation of antigen to CD8 plus T cells. These are the presentation of antigen to CD8 T cells. See, these are the MHC class first molecule. These are the alpha chain which cross this transmembrane, right? And these are presented on the CD8 T cells. Next is MSC class 2 molecule. These are consist of alpha chain and beta chain, right? These are consist of alpha chain and beta chain, both anchored in cell membrane. Both anchor the cell membrane and both are polymorphic. Polymorphic means already I told you that they are present in different form, means like alpha 1 and alpha 2, beta 1 and beta 2. So alpha 1 and beta 1 domain form peptide binding cleft. Alpha 1 and beta 1 domain form peptide binding cleft. In MSC class first molecule, only alpha 1 and alpha 2 form the peptide binding cleft, whereas in class 2 molecule, alpha 1 and beta 1 form a peptide binding cleft. Expressed on professional antigen presenting cells. Present on professional antigen presenting cells, that is APC, such as B cells, dendritic cells and macrophages. Ligand for CD4 and TCR, presentation of antigen to CD4T. Ligand for in class first molecule, what happened? They are represented on CD8 T cells. In class 2, they are presented on CD4 for T cells. Peptide binding group of MSC class first molecule. Alpha 1 and alpha 2 domain form peptide binding domain. Anti-parallel 8-stranded beta pleated sheet form the floor. What happened? Anti-parallel 8-stranded beta pleated sheet form the floor. Two long alpha helix oriented adjacent and roughly parallel to each other form side of the deep cleft. Closed ends accommodate about 8 to 11 amino acids. What happened? They accommodates about 8 to 11 amino acids in a flexible extended conformation. So peptide binding to class first MSC. See, this is the peptide binding to class first MSC. Solvent accessibility about 80% buried. Size does matter, like 9 mol best. So peptide binding group of MSC class. Peptide binding group of MSC class second molecule. Two long alpha helices are sides and the beta sheets are as bottom. The ends of class 2 peptide binding cleft are more open. What? They are more open. Binds longer and irregular peptide. Means in this what happened? 12 to 16 amino acids of the peptide. In class first molecule what happened? The class first molecule are only expressed in nucleated cells, right? And in class first molecule approximately 8 amino acids are recognized in class first MSC, but in class second MSC they recognize some longer. They bind some longer amino acids of peptide, right? Like 12 to 16 amino acids. See here, this is the MSC2. Longer peptides ends can hang out central core of about 13 amino acids. That is allele a specific binding motif. This is the peptide that bind to this MSC class 2 molecule. This is the 16 or 13 amino acids long peptide that bind to this MSC class 2 molecule. Now these are the difference between the class first and class 2 MSC molecule. Peptide binding domain alpha 1, alpha 2 in class first molecule, in class 2 molecule alpha 1 and beta 1. Nature of peptide binding cleft, that is closed at both ends, that open at both ends, right? Nature of peptide binding cleft. General size of bound peptide in class first is to 10 amino acids and in class 2 13 to 18 amino acids. Peptide motif involved in binding to MSC molecule, that is anchor residue at both ends of peptide, generally hydrophobic carboxyl terminal anchor, and in class second molecule anchor residue distributed along the length of the peptide. Nature of the bound peptide, extended structure in which both ends interact with the MSC and cleft, but middle arc is up away from the MSC molecule. Whereas in class 2 molecule, extended structure that is held at a constant elevation above the floor of MSC cleft. MSC peptide interaction. There are about a dozen of types of classical HLA molecule on the cell surface for one individual. However, there are much more kinds of antigen peptides would be presented in one individual, right? So the main function of HLA molecules are first are the antigen presentation, that is process the endogenous antigen and exogenous antigen. Exogenous and endogenous antigen. So presentation are T cell recognize MSC peptide complex, MSC restriction of T cells. Any individual T cell respond to a specific MSC allele expressed by that individual, that is to self MSC. What? Any individual T cell respond to a specific MSC allele expressed by that individual, that is to self MSC. and other is genetically regulator of immune response, so to predispose individuals to particular susceptibility or disorder. Particular susceptibility or disorder, immune regulation. They also help in the immune regulation. Now, there are feature of the MSC gene. First is polygenicity, second is polymorphism, third is haplotype inheritance, fourth is linkage disequilibrium, and fifth is codominant expression. These 5 are the feature of the MSC. So, we discuss individually. First is polygenicity. So, what are the polygenicity? In polygenicity, multiple separation and functional gene encoding the similar MSC. What? Multiple separate functional gene encoding the similar MSC, that is HLA first, in HLA first, HLA A, B and C. HLA second, HLA DR, DP and DQ. See here. HLA first, in HLA first what happened? They are encoded by A, B and C, right? This represent the alpha chain. Clear? A, B, C. This represent alpha chain. In second what happened? DR, DQ and DP. DR, DQ and DP. This represent alpha and beta both chain. So this both MSC first and in MSC second, in both these are represented by multiple gene, multiple functional gene, right? So that's why it's known as polygenicity. That's why it's called as polygenicity. Next is polymorphism. Polymorphism means there are many different alleles present among the different individuals in a population. Polymorphism means there are different recognition, or they are present in different form. Like alpha chain are present means their different form are alpha 1, alpha 2, alpha 3. If beta chain are present, they are different form of beta chain are beta 1, beta 2, beta 3. So alpha and beta both are present in different form. This is known as polymorphism. Most polymorphic genetic system known in higher vertebrate. These are the alpha 1, alpha 2, alpha 3, beta 2. These are the polymorphism. Same gene are having the different form, like alpha 1, alpha 2, alpha 3. Right? They call alpha chain, and this alpha chain are separated in alpha 1, alpha 2 and alpha 3. This represents this polymorphism. Now haplotype inheritance. Haplotype means one gene comes from one parent, another comes from another parents. So this type is known as haplotype. The specific set of allele for all MSC loci on one single chromosome. Usually, in fact, haplotype are passed on to the next generation. One chromosome are passed to the next generation. One from parents and one from mother. So haplotype are passed from to the next generation, right? So this is the haplotype inheritance. Most people are heterozygous at HLA loci. Linkage disequilibrium. Certain MSC allele at different loci are inherited together more or less frequently than would be predicted by random assortment. HLA A1 that is 60%, HLA 2, HLA B8 9%. So the representation or presentation of the gene or linkage of the gene are different in different type of gene. Like alpha 1 are linkage of alpha 1 at 16%, linkage of beta 8 are 9%. So the linkage of different gene are different. So this is known as linkage disequilibrium. There is no equilibrium. There are changes. Now codominant expression. Codominant expression means both the allele are presented at the same rate in same individual or at the same moment, right? in like a blood group, A and B both present their quality both present their quality equally. So blood group are a co-dominant phenomena. Like that only, in this also MSC are the feature of MSC like a co-dominant expression. Means both the gene of the maternal and paternal that are inherited are expressed equally in a cell. That is a co-dominant phenomenon. So main feature of MSC are providing the strongest barrier to transplantation, play a central role in generation and execution of immune response, presentation of peptide antigen to T cells, and susceptibility to infectious disease and development of autoimmune disease. So the main function of the MSC are in the transplantation or in the grafting of the organ, right? So this all about the HLA, MSC that are present in the body. So MSC are of two types. Mainly there are three types: MSC first, second, and 3. And this MSC are located in a chromosome number 6 in human, right? So hope this video are clear. Today we stop here. Thank you.